Hi viewers, welcome to SEKHAR'S Bio Factory for NEET. If you like this video, subscribe my channel and click on bell icon to get notification. In this video, I am going to explain one of the topic in cell cycle and cell division chapter. It is mitosis division and its mechanism. The term mitosis, it was first time given by one of the scientists, Walter Fleming, in 1882. But the process of mitosis, it was first time observed by Strasburger in case of plant cell and Walter Fleming in case of animal cell. In mitosis division, the mitosis division is a process. In this one parental cell, it can divides into two identical daughter cells, and these two daughter cells are identical to each other, and also they are similar to its parental cell. During this mitosis division, the chromosome, it consists of a DNA, and this DNA, it is duplicated in S phase, and this duplicated DNA, it present in the form of chromatid in chromosome, and this duplicated DNA, it is equally distributed in two daughter cells in case of mitosis division. And mitosis division is also called as equational cell division because when we compare the chromosome number in both parental cell and daughter cells, so both parental cell and daughter cell, it consists of same number of chromosomes without any change. That's why mitosis division is also called as equational cell division. This mitosis division, it frequently observed in somatic cells. That's why it is also called as somatic cell division. In case of plants and animals, the body cells perform growth with mitosis division only. In case of plants, the mitosis division is the main characteristic feature of a meristematic tissue. And this mitosis division, it also occur in gonads. So in gonads like male reproductive organ like testis and female reproductive organ like ovary, in these gonads to multiply undifferentiated germ cells, mitosis division takes place. Along with that, in plant body, the meristematic tissue is the main site for mitosis division. Along with that, mitosis division can be seen in young leaves, flowers and fruits for growth. Mitosis division, it is an elaborated process because during this mitosis division, a series of changes can take place in both nucleus and cytoplasm of the cell. So there are two important major events occur in mitosis division. In this, the first event is karyokinesis. Second event is cytokinesis. In case of karyokinesis, the nucleus, it divides into two daughter nucleus. During this process, all changes, it occur continually to form a two daughter nuclei. And these changes in karyokinesis further classified into four stages. In this, the first stage is prophase, second one is metaphase, third one is anaphase, and fourth one is telophase. In case of prophase, in prophase, pro means first, phase means stage. When the cell, it enters into M phase during cell cycle, when it enters into prophase, we can observe some important features like condensation of chromatin material into chromosomes. Along with this, during this condensation process, the DNA strand, it gets untangled to form compact mitotic chromosomes. Before prophase, the DNA which present in chromatin material it is present in the form of network. And this network formed chromatin material is untangled that can remove that network, then finally it becomes into compact mitotic chromosomes in prophase. Along with this, we can observe one more change in cytoplasm in case of animal cells, because in animal cell cytoplasm during S phase the centriole duplication take place, and these duplicated centrioles they move towards opposite poles in prophase itself only. Moving these centrioles towards opposite poles is leads to polarization. The chromosomes in this case, it consists of two coiled sister chromatids, but these two sister chromatids are joined with a structure, it is called centromere. In this prophase at beginning or at early prophase, the chromosomal ends are not clearly visible, and initiation of assembly of mitotic spindle fiber it occur itself in prophase, because in cytoplasm of animal cell the microtubules, these are the proteinaceous components, and these microtubules these are assembled into spindle fibers, and these spindle fiber formation initiated in prophase phase itself. At the end of this prophase, in the structure of a cell, when we observe under microscope, at the end of prophase, we can observe the disintegration or disappearance of nuclear membrane, nucleolus, and along with Golgi complex and endoplasmic reticulum, it is seen at the end of prophase. Coming to the second phase in mitosis division in karyokinesis, it is metaphase. So when the cell after completion of this prophase, then it enters into metaphase, and when it enters into metaphase, at this time the nuclear envelope and it completely disappears. The chromosomes which are present in nucleus now it spread freely in cytoplasm of the cell at beginning. So condensation of chromosome, the process is completed when the cell enters into metaphase, and at this time the chromosomes are very clearly visible under microscope. In the structure of each chromosome, it consists of two sister chromatids, and these two sister chromatids are attached with the help of centromere, and in this centromere we can observe some small disc-shaped structures around the centromere, and it acts as a site for the attachment of spindle fibers, and is called kinetochores. The chromosomes in this phase they become very short and thick, and this metaphase is the best phase to study the morphology and structure of chromosomes. The chromosomes it consists of it is condensed into a short rod-like form in metaphase itself. The spindle fibers, these are made up of microtubules, and this mitotic spindle fiber formation process is completed in metaphase, and these mitotic spindle fibers get attached to kinetochore of centromere. The spindle fibers, when it attaches to kinetochore of centromere, they can bring all these chromosomes at the middle of the cell, it is called equator, and bringing the chromosomes at equator is the process called congregation. And all chromosomes now align themselves at equator. The plane of alignment of all centromeres in chromosomes, and it leads to formation of a plate-like structure, and it is referred to as metaphasic plate. So all centromeres forms a metaphasic plate in metaphase. The chromosomes in its structure. These are aligned at equator. At this time, one of the chromatid it is present at one side with the kinetochore and spindle fiber. Another chromatid present at opposite side with the help of kinetochore and spindle fiber. Both are present at opposite poles. In case of anaphase, it is the third phase. In anaphase, the chromatids they start movement towards opposite poles. When the cell it completes metaphase, then it enters into anaphase, and this anaphase is characterized by spindle fiber attachment to kinetochore. And in this anaphase, these spindle fibers it becomes shortened, and that means the length of each spindle fiber decreases, and these spindle fiber it create pressure on centromere, and due to this pressure the centromere it is splitted into two centromeres, and then the two chromatids now it is splitted into daughter chromatid, and it is referred to as daughter chromosomes. So these daughter chromosomes they start their movement or they start migration towards opposite poles with the help of spindle fibers, and it is seen in anaphase itself. What are the chromosomes separated? And these separated daughter chromosomes they move away from equatorial plate. In anaphase at their end, the daughter chromosomes they reach opposite poles. This anaphase is the best phase to study the shape of chromosomes based on the position of centromere in the structure of chromosome that may appears like either L shape, V shape, J shape or I shape depending upon the position of centromere in chromosomes. The last phase of karyokinesis is telophase. When the cell enters into telophase, and it is the end stage of karyokinesis in mitosis division. At this time, the chromosomes it consists of two sister chromatids. They reach to opposite or their respective poles, and the mitotic spindle fibers are completely disappear at the beginning of telophase. In this telophase, the decondensation of chromosomes it take place, then these chromosomes finally it becomes into indistinguishable mass and collect at opposite poles. In this way, the chromosomes it will be decondensed into a network form chromatin material at opposite poles. During this telophase, one of the most important change it occur in telophase, it is the chromosome it lost its identity, because the chromosome is not appeared now, it is appeared in the form of a network form chromatin material. At the end of this telophase, the nuclear envelope it assembles around the chromatin material or cluster. Along with the nuclear membrane, nucleolus, endoplasmic reticulum, Golgi complex, and other cell organelles are also reappeared in cells. So, the second main phase in mitosis division is cytokinesis. In cytokinesis, mitosis division not only concerned with division of nucleus, along with the nuclear division, cytoplasmic division also takes place. It is referred to as cytokinesis. And the division of cytoplasm it occur after completion of karyokinesis is referred to as cytokinesis. After nuclear division, karyokinesis, the cytoplasm of the parental cell it finally divides into two identical daughter cells. It is a change seen in cytokinesis. After cytokinesis, the two daughter cells they have their own nucleus and cytoplasm in its structure. During this karyokinesis, the cell organelles which are present in cytoplasm of the cell like mitochondria, plastid and remaining cell organelle, all these are equally distributed in two daughter cells at the time of cytokinesis. The cytokinesis, the process is different in different cells like plant cell and animal cell. In case of animal cell, the cytokinesis process takes place with a method. It is called cell furrow method. In this cell furrow method, in case of animal cell, in animal cell, due to absence of cell wall around the cell, the cytokinesis is achieved by a formation of a furrow in between two daughter nuclei with the help of microfilaments of cytoskeleton. The furrow which is formed in plasma membrane, it deepens. That means the movement starts from peripheral and the deepened or furrow it moves towards the center. It is the growth called centripetal growth, and ultimately this furrow can be joined to divide the cell into two daughter cells. It is the process called cell furrow method seen in animal cell. The second method of cytokinesis, it is called cell plate method. In this cell plate method, it is seen in case of plant cell because plant cell it is surrounded by well organized cellulosic cell wall. In plant cell, the cytokinesis process is achieved with the help of cell plate method, and this cell plate it represents middle lamella in between two neighboring or adjacent cells. So during this cytokinesis in plant cell, first with the help of Golgi complex, it forms a structure in between two daughter nuclei. It is called as phragmoplast, and this phragmoplast it grows from center to peripheral that leads to the formation of a plate. It is called cell plate. During their growth, the growth of phragmoplast takes place from center to peripheral. It is called centrifugal growth. And this cell plate, it starts from center and move towards peripheral centrifugally, and then it can divide the cell into two daughter cells by forming a wall in between these two daughter cells. It is called cell plate method. But in some of the cells, very rarely, only in few organisms. So karyokinesis it is not followed by cytokinesis. That means in a cell the nucleus divides into two daughter nucleus, but it delay or prevents cytokinesis process. Finally it results in the formation of a many nucleus in a cell that leads to multinucleate condition. So the process of formation of multinucleated condition due to failure in cytokinesis is referred to as syncytium. Best example for syncytium, it is a liquid endosperm in coconut. The coconut water, it is nothing but liquid endosperm, and this liquid endosperm is composed with many nuclei along with cytoplasmic content. But during maturation, every nuclei it can synthesize cell wall around it, then it becomes into solid coconut or solid endosperm. But the liquid endosperm it is example for syncytium because it consists of a multinucleate condition. Thank you for watching. If you like this video, subscribe my channel.